The paper we're discussing today presents a new way to measure how different parts of the brain work together during thinking and memory. Instead of focusing only on whether the brain signals fire at the exact same moment, the researchers here examine how the strength of rhythmic brain activity, as in electrical brain waves, rises and falls across groups of signals over time. They develop a mathematical method where they can detect shared patterns in very complex brain recordings, even when many signals are involved at once. After testing the method in stimulations, for example, and applying them to real brain data during the memory task, for example, they show that it can reveal meaningful coordination between different brain regions. And so this study is significant because it offers a clearer and more scalable way to understand how distributed brain areas and brain regions communicate during cognition. To understand why this matters, it helps to step back a little bit and consider how the brain communicates internally. The brain is not a single processor, so to speak, but a distributed network of specialized brain regions. These brain regions exchange information through electrical activities that often takes the form of a rhythmic neural oscillations. And neuroscientists have long suspected that these oscillations help coordinate communication, but measuring coordinate across many signals simultaneously is technically and mathematically quite challenging for neuroscientists. And so traditionally, these researchers examine the synchronization by looking at phase relationships. In simple terms, phase refers to whether two oscillations are aligned in their peaks and then in their less peaks, so to speak. If two signals rise and fall at the same time, they're said to be phase synchronized. Now, while this is useful, it captures only one dimension of coordinations between these neural networks, and so oscillation also vary in amplitude, meaning that their strength increases and decreases over time, and these fluctuations can carry important information among all these brain regions. Even if two oscillations are not perfectly aligned in this phase, their amplitude may increase and decrease together over longer timescales, and this type of coordination may reflect functional interaction between brain areas engaged within the same task. And the difficulty is that real neural data often involve dozens or hundreds of simultaneous channels, and making pairwise comparison is insufficient and statistically unstable, so to speak. And so the researchers here in this paper address that this high dimensional challenge by extending canonical correlation analysis, which is a classical statistical method used to find relationship between two sets of variables. In its traditional form, canonical correlation analysis identifies linear combination of variables in one set of data that best correlate with linear combinations in another. However, applying this method directly to neural time series is problematic per se because neural data are extremely noisy. They're also temporally structured and often high in dimensionality relative to your number of observations. To help overcome these mathematical issues, researchers here developed a specialized framework tailored for oscillatory time series that happens in brainwave activities. They extract amplitude envelope from neural signals of these brains, and they constructed structured covariance models that accounted for temporal dependencies. And so by doing so, they identified dominant patterns of cross population amplitude coupling rather than relying on just these unstable pairwise comparisons of neural activities. And one important innovation here is that in their approach, these researchers emphasize on dimensionality reductions of these neural networks instead of asking whether every channel in region A of the brain coupled, for example, with every channel in region B. This method identified collective patterns of these neural networks, and it found weighted combinations of signals within each region whose amplitude envelopes exhibited maximal correlation with combination in other regions of the brain. And this reduced the complexity while preserving the interpretable structure of the data and the computation itself. Researchers in this paper validated their method using simulations in which the true coupling structure was known. In these controlled scenarios, for example, the algorithm reliably recovered the embedded pattern of amplitude coordination, where this step was extremely crucial because it demonstrated that the framework can distinguish real structured interactions from random fluctuations. And after confirming its performance in simulated data, these researchers applied the method to neural recording obtained from local field potentials, which is the brain activities deeper in the brain, more deeper than EEG, in two different brain regions, one in the prefrontal cortex and one in the visual areas V4 during a working memory task of the participants. These regions are primarily known to interact during memory maintenance and visual processing of the individual, and the goal here was to test whether cross population amplitude coupling could reveal plausible communication patterns during cognitive engagements of these regions. The analysis here revealed structured amplitude correlations between two brain regions. Rather than random or diffuse interactions, the method here identified specific low dimensional modes of coordinated amplitude fluctuations. These modes likely reflect functional channels of communication that become active during the task. And so, conceptually, this work supports the idea that communication in the brain may be mediated not only by precise spike timing or phase locking, but also by coordinated modulations of oscillatory strength. When the amplitude, for example, of a rhythmic brain signal activity increases in two brain regions at the same time, it may signal that both brain regions are engaged in a shared computational process. And this interpretation aligns with broader theories of oscillations and communication through coherence within cognition and within these brain regions. From a methodology perspective, the framework here provides a more scalable way to analyze modern neural data sets. As recording techniques advance, neuroscientists can measure activity from increasingly large populations of neurons and channels. Analytical tools must then evolve accordingly. Methods that can rely on exhaustive pairwise comparisons quickly become impractical and statistically fragile, and grounding the analysis in established multivariate statistics while adapting it to the structure of neural oscillation. Researchers here created a bridge between classical data analysis and modern neuroscience challenges, and the resulting model is not merely a black box, but a very structured decomposition of interregional couplings within the brain. This method is also could be very generalized. For example, here the researchers demonstrated that on local field potential, the sensors that are invasive within the brain, from two specific cortical regions that we mentioned, prefrontal and visual, the methods here can be applied to other types of neural recordings, for example, EEG, MEG, and so forth. It could also extend beyond neuroscience to any domain involving high dimensional oscillation system. And so for the intellectually curious readers or viewers without deep neuroscience training, the broader takeaway here is that understanding the brain increasingly requires sophisticated mathematics. The brain's complexity cannot be captured by simple measures of synchronization. Instead, meaningful patterns often emerge only when we look at coordinated structure across many signals within the brain simultaneously. And in the context of cognitive sciences, this study reinforces the view that memory and perception arise from distributed network dynamics rather than isolated brain regions. Coordination is not binary, so to speak, but graded and structured, and amplitude envelope coupling provides one window into this subtle orchestration of the brainwaves activity that happens within brains. Ultimately, this paper contributes to an evolving picture of the brain as high dimensional dynamical system whose components communicate through pattern fluctuation in electrical activity, and by offering a principled way to detect and interpret these patterns of oscillation, researchers here advance both at the methodology level, but also at the conceptual understanding level, where the work demonstrates that when statistical innovation meets neuroscientific theory, new forms of insights into cognition become increasingly possible. The framework introduced in this paper also aligns very naturally to the core idea of symbiotic intelligence, which views intelligence not as a property of isolated units, but as an emergent phenomena arising from coordinated interaction across biological and artificial systems. And so, for us to identify low dimensional modes of amplitude coupling between brain regions, the study shows that meaningful cognition depends on structured relationship rather than independent activities of the brain regions. In the language of symbiotic intelligence, for example, these amplitude correlations can be understood as signature of cooperative integration, where brain regions and brain areas are not merely exchanging signals, but they are dynamically aligning their levels of engagement with one another. And so the intelligence of the system emerges from this alignment where pattern co-modulation that allows distributed brain regions to operate as a unified process while retaining its local specialization within each brain. And the mathematical and statistical method presented in this research also reinforces a system level perspective that is very central to symbiotic intelligence. If cognition depends on coordinated amplitude fluctuations across neural population, then intelligence can be conceptualized as a network property shaped by mutual modulation and shared dynamical state. So this supports the idea that biological intelligence is inherently relational and that higher order capabilities arise from structured coupling across different components. In applied contexts, for example, such as human machine integration and interactions, or future neuroadaptive systems, detecting and enhancing these coupling modes could become a pathway toward deeper symbiosis between these artificial and biological systems. And so this study, therefore, provides not only a technical advance in computational neuroscience, but also an empirical grounding for the principle that intelligence scale through structured cooperation across interacting systems, be it a biological system, whereas an artificial system. And that's all I had for today. Thanks for tuning back in. I see you guys next week. Bye.